A very good evening to everybody. Welcome to another episode of Eclectica, the different news. Today, I have a very interesting guest with me. I welcome Dr. Saurabh Bhattacharya, the Director of Cytogenetics Department at Freiburg Medical Laboratory at United Arab Emirates. Welcome, Saurabh, and thank you
Thank
so
you.
much for accepting my invitation and coming on my podcast.
Thank you. My pleasure.
Thank you, Shorav. And I think a very interesting area. Personally, I am very inclined to the field of genetics and cytogenetics. I think it will be a very inspiring, insights that we will be getting from you today. So before I go ahead, please tell us about your journey, how you reached in this space, and because I think I'm sure it would be a very exciting journey that you have had in the past and you're ongoing. So please tell us about yourself.
Yes. So about me, I born and brought up in Madhya Pradesh, very small place in Madhya Pradesh, Naurajabad, where I born and did my schooling. And I'm sorry, I'm going very back, but I just wanted to share that how I started this journey. And then I moved to a place called Jabalpur. It's a well-known city in Madhya Pradesh where I did my graduation. My graduation was in biochemistry, and my major was biochemistry and then zoology and chemistry. From the beginning, I was very much interested in the chemistry subject because I was very fascinated about the chemical bonds and different reagents and everything, especially inorganic chemistry. Because I believe that if you have a very interesting and good teacher, you will love that subject. And that is what happened to me. And of course, my school, I always choose biology as my major subject. I was very bad in maths, so I just escaped from there, and then I did my graduation. But in how I got interest in genetics was because in the zoology there was one small chapter in our subject in our curriculum when my teacher she started teaching me about the chromosomes that time. So it was very fascinating for me that how that chromosomes can carry a genetic information, and it tells us that what we are going to be and how we behave and everything, the genetic traits and everything. So I got interest on that, and then of course biochemistry was my kind of a favorite subject in chemistry, but I moved my area to zoology, especially in the genetics. But chemistry is always important in the genetics part because everything is linked with the bonds. We know about the DNA, it is how it linked. So I was thinking that where I will pursue my genetics. Then I was keep on searching, and then I did not find any particular college in our state because Madhya Pradesh that time it was like I'm talking about 2002. It's like 22 years back, and very few people are really interested in genetics part. Everyone wanted to go for they're doing their masters or do their MBA or something like that. But I was focusing on that, and then luckily I got an admission in Bangalore. So there is a university in Bangalore University. I got an admission, but it was very expensive that time, and financially we were not very strong. My father was very much, keeping stressful, and then he was thinking that what we do and how we manage the fees and everything. So he was on the way on the train, and he was coming back to Madhya Pradesh after getting the token money for the admission. On the way in the Vellore is a Tamil Nadu place where you must have heard about Christian Medical College there. One gentleman got up in the train and he was going to Bihar. And he was speaking to my father and said that why don't you try Vellore Institute of Technology, which is very well known university. And my father said, okay, let's talk to my son and see if it is really feasible for him. And I never heard about that university in MP because that time it was like very new and it was known for the engineering college only. And he asked me whether you were interested. I said that time no internet, proper internet in our place, and no phone, landline and all. So he called me from one station, and then he told me that I met someone and he is proposing this university, and his son is also studying there. If you are interested, we should come back, and then we check that. I was kind of a depressed because I was thinking, okay, I will go to the Bangalore and I will study there. All those things in a young people who have thinking about Bangalore is a very very fascinating place that time, and especially I'm from the very small place. I was thinking about that. So, but when my father came back and then he said, okay, let's go and find out this Vellore Institute of Technology. Trust me, when I entered the university, I was in a stars because I never saw a beautiful campus like that. Luckily, I got an admission in biomedical genetics subject. I got an admission, and only one seat was available. Maybe God wanted me to go there or something. I don't know. And that person who met my father has become like my godfather that time because he was the one who proposed. And then I started my journey in genetics there. And slowly, I was not very good in English because I was from Madhya Pradesh. We are not talking much English there. I had lots of difficult time there because everyone is very smart. The students were talking so nice English, and the teachers, South Indian accent and all, very difficult for me. So somehow I finished my first year, and then when I entered my fourth semester, yes, when I entered my fourth semester, there was a curriculum called as cytogenetics, a particular special subject for cytogenetics. There I found my passion, and I started doing my best in. I was not very good in other subject, biostatistics and different other subjects, but I was very good in cytogenetics, and I took it as my passion, and I started looking at my chromosomes and doing it. I was kind of very much fascinated. Then the last semester was for the project, 6 months project, and people were running here and there. Few people were going to USA, few were going to Australia to do the project. Some people going to some other companies, and I was the only one who left behind because nobody was choosing cytogenetics. They were going for molecular or some kind of cancer related molecular projects and everything. But I told my guide that no, I will do this cytogenetics if you allow me to work in your lab. So I stayed back in VIT, Vellore Institute of Technology. I was the only one out of my whole batch that who stayed back in university. Everyone has gone out. People were saying, yes, I have faced lots of things that people were saying that what will you do in cytogenetics? There is no future and everything. I said, okay, let's follow my passion. Let's see. And I feel that if you follow your passion, if you love your subject, you will do someday. You will do better, and that's what.
Absolutely.
Yes, and that's what I did. I did my project, and my project was very interesting. I worked on the post polio syndrome, and I found the novel chromosome aberration which was never been detected before, and it was very good project. Then my guide has proposed me that okay, you continue your research in this. I will propose that you can become my student, and I will guide you. I said, okay, I will be ready. But of course, financial constraints were always there because I was studying and I wanted to help my father. But I got a scholarship also in the university, and then I supported my study and I was helping my father as well. Then I continued my research, and in 2010 I finished my research, and it was published and everything. I got a proposal for postdoctoral fellowship also from Karolinska Institute because the person pioneer in this field, Dr. Ken, he asked me to join his institute and everything. But I was very, thinking. I was in a place where to choose a career or to choose a profession and then earn the money so that I can support my father and I can take care of my education loan, which I have taken for my studies as well. So I choose to stay back in India and try to continue my work. And luckily, in 2012, I got an offer from Christian Medical College to join their cytogenetics department because I wanted to get a diagnostic experience. So, I had a very good research experience, but I wanted to get a diagnostic experience. And there, I got a chance to work as a senior demonstrator because that time I did not receive my degree yet. So and they told me that if you once you get your degree, we will promote you. And that what happened in 2012. I got my degree finally, and then I was promoted to the senior lecturer and then tutor PhD. And I was taking some classes for cytogenetics for especially MD pathology students who were coming for fellowship. And I was working as a diagnostic scientist kind of designation in the department of cytogenetics. I got lots of exposure in cancer that time. Then I moved to USA and Germany also for my further training in cytogenetics. I got training in fish fluorescence in situ hybridization, which is very commonly used now. and then microarray. And after 1 year, 2013, I got an offer from India's largest diagnostic lab, Dr. Lalpath Labs, and I was invited for interview. And trust me, that was the first time I got into the aeroplane from Chennai to Delhi. And yes, I was there, and then they selected me, and I started my journey in 2013 in Dr. Lalpath Labs. And till I come to Dubai, I was working with Dr. Lalpath Labs as a director of cytogenetics. I started my journey with the senior scientist and then slowly to head of the department, national head, and then director. And when I came here, I also got an offer from Dubai, Freiburg Medical Lab. So they asked me to establish their department. It was a very big challenge for me because it was completely empty floor for me to establish a lab here. And now where I'm sitting, this is what I designed here, and then we started. And now in Dubai, Freiburg Medical Lab Cytogenetics is the only department who does cytogenetics, and no other labs, because everyone is sending samples outside, but we are the one who is doing, and we are doing pretty well. This is what my journey is, and that's what I am.
Amazing. And I think I was just boiling inside with excitement when you were telling your journey. Though it is your podcast, it's all about your journey, but I'm excited to share the connects. I think it's very similar. My first for the interest in genetics stemmed for myself also was zoology, because
Uh,
my first bachelor's was like, even biochemistry is something which I love till date. I can talk hours together. And I studied in Bangalore. In
oh,
Bangalore, my postgrad was in applied genetics. And
mhm.
the first time when I, like, it was like our in-house student project. So our department HOD was, he used to work in a, like, we used to laugh as students, but I understand the value today. He used to work on mosquitoes. We would like, what? Who can work on mosquitoes? The
Yeah,
first time I isolated Lambrush chromosomes from the salivary gland of mosquitoes, and when we
wow,
saw on the microscope, under the microscope, as a student, it sparked so much. So I
right.
think, I mean, your journey is very, very inspiring, and thank you so much for sharing that. Um, so I think, Saurav, people from the background of genetics, we know a lot about cytogenetics, but let me ask you with a very ABCD. So can you explain, mostly for all of us, for my audience here who might not be from the field of genetics, so can you tell us in a simple way what exactly cytogenetics is, and probably what is the importance it carries in when it comes to medical diagnostics?
Okay. So as cytogenetics is made up of two words, cyto and genetics. So we are studying, cyto means cell and genetics is the study of genes, right? So what we do in cytogenetics is we study chromosomes. And these chromosomes can be studied with different other technologies. For example, karyotyping, and then fluorescence in situ hybridization, and then microarray. These are the major three technologies which is used in cytogenetics. But of course, there are subtypes of other technologies which are linked with this. For example, if I talk about karyotyping, we culture the cells, blood cells, and then we do some harvesting and everything, and then we make the slides and see under the microscope the chromosomes are beautifully elongated chromosomes. But if I want to paint the chromosomes, I will use some probes and do the painting in the chromosomes, and then I see the colored chromosome. That is kind of a fish, fluorescence in situ hybridization, or we also called it as spectral karyotyping. So these are the things which we do in cytogenetics. And then microarray. Microarray is a bit different from these two technology, because in microarray we extract the DNA from the patient sample. It can be a blood sample, it can be a, tissue sample, it can be salival sample. We just extract the DNA and then run in the chip and see in a whole genome analysis. We see the chromosomes abnormality in a better way. So that is what a cytogenetics is. So cytogenetics is grossly detecting a copy number variants, whether it is a duplication, deletion, translocation, structural variants, and the loss of chromosome, gain of chromosome, all those things. So these are the broad technology which is used in cytogenetics. Most widely, karyotyping is used because karyotyping is a gold standard still. We can see all the 46 chromosomes all together in one metaphase. and we can see whether gross abnormalities are there or not. But of course, all the technology has their own drawbacks, limitations as well. So what karyotyping cannot detect, fish can detect. We can take the probes for the particular gene and we see whether there is a deletion or no. Because sometimes karyotyping has a resolution constraint, then we cannot see the deletion, micro deletions. We can detect in a fish. But if you want to see all together all the chromosomes and with the better resolution, microarray is the best test. So these are the major technologies used in cytogenetics. And then if I talk about what is the use of cytogenetics in a medical field and medical genetics, if I say cytogenetics is very important because, for example, if I take the cases of infertility. So in infertility, there are different types of infertility, primary infertility, secondary infertility. So in if I talk about secondary infertility, it is very easy to understand that couple are trying to get a baby. They get a baby, then baby is getting aborted, or during the pregnancy there is some problem and all. So we need to check whether these couple have some kind of chromosome abnormality or not. Because if they are carrying the balanced translocation in any of the chromosome, the chances of getting a fetus with abnormality is 50%, right? So that's why we have to do the cytogenetics basic test to be done here. But there are certain other tests like during pregnancy, you can do now very commonly done NIPT or NIPS, a non-invasive prenatal test or prenatal screening, where you take the mother's blood and see the chromosome aneuploidies, like the trisomy 13, 18, 21, and so on. And then that is screening test. Once that is confirmed that the patient is carrying, the fetus is having a trisomy 21, for example, the best thing to confirm is through cytogenetics. So the patient will go and visit their gynecologist. They will take the sample in the form of amniotic fluid through amniocentesis or chorionic villus or cord blood sample, depending on the pregnancy age of the pregnancy. And then that sample will come to the lab of cytogenetics lab. We culture the cells and then take out the chromosome and confirm that there is a trisomy 21 or trisomy 13 or maybe some other abnormalities which is still there, which NIPT cannot pick up. So that's where cytogenetics is very important. So I talked about infertility and then prenatal diagnosis. Then there is a couple where they have a recurrent abortions, 2 abortions, 3 abortions, more than 4 abortions. Even in my career, I have seen a people with 8 to 9 abortions as well continuously, and we found that there is a balanced translocation in either of the couple. It is not necessary that male or female, male will carry a translocation or female will carry. Either of the couple can carry, and when they have a haploid chromosomes, it will go in the fetus. There are chances of having abnormal fetus or an abortion because the fetus may carry that abnormal chromosomes. That is what it is. So here it is I spoke about prenatal and postnatal. Now cytogenetics is also important in cancers. So if I talk about the hematological malignancies, leukemia, for example, very classical example is chronic myeloid leukemia. We see Philadelphia chromosome 922 translocation. So first thing to do when patient comes to the doctor and doctor suspects the patient may carry leukemic cells in his body, the first thing to do is the karyotyping. Because we may get complex karyotype, we may get 922 translocation, we may get 821 translocation, different types of abnormality which is directly associated with the disease outcome. So here cytogenetics is very important. And then when patient goes for therapy, because this is not this is somatic abnormality, right? So patient can get a therapy and then the abnormal cells may go out and repair in the body and they come out. So to follow up, fish is very important. So which we have detected in cytogenetics earlier, we can follow up with the fish. We can use the probe and see the percentage of cells in the body of the patient, how much percentage is there, still there or not there, or completely remission has been achieved. So these are very important in cytogenetics. And then if you talk about the breast cancer, very classical example, breast cancer, HER2 gene or Herceptin gene, which is very important to detect if there is an amplification that can be done by fish. So fish is the gold standard for that. Take the tumor cells, do the processing and do the fish and see the amplification. If there is amplification, then we see as per ASCO guidelines, if it is a higher side or the intermediate side or the lower side, then we can give the report and patient can get benefit through Herceptin therapy. So broadly, I explained that the cytogenetics is how it is important in not only in prenatal, postnatal, it is important in cancer as well. So that is where it is really important to do cytogenetics. Yeah.
Thank you for that extensive explanation in a very simple way. I understand these are quite, in terms of science, it is complex, but you gave us a very broad umbrella overview of the entire aspect. Thank you, Sora, for that. So, like as you said, I rightly that karyotyping and the technologies inside chromosomal analysis has undergone a lot of advancements at this point. Like fish, we have sky and
Yeah.
like spectral typing, and like as you said that comparative genome hybridization, microarray and
Right.
stuff. So what exciting I'm more, my question is more towards it's from a clinician's perspective. So what exciting developments do you foresee in this field more in the coming years?
Okay. The major advancement what I see in the coming years is the implementation of artificial intelligence. So here we say that okay, cytogenetics is completely manual process and everything, and it needs expert eyes. But of course, now I have seen that there are certain companies which are coming with the artificial intelligence. So they help a cytogeneticist to come to the conclusion very precisely. The softwares are available which are doing the karyotyping also with 98 to 99% accuracy. But of course, how they do that, because if you have to give a quality chromosomes to the software. If your chromosomes are really good and quality is good, you have a proper banding and everything, the software will really help you to detect the abnormalities and help you to understand. But of course, as I say, no one can replace a human brain, right? Because artificial intelligence is also made by human. So it will help you, it will give you an indication that there is a abnormality, but as an expert you must decide that whether it is really a reportable abnormality or it is just an artifact or something like that. So that is where I would say the advancement is going. But now in terms of technology, there are so many robotic microscopes, so many robotic slide preparator. Even in India, when I was working with Dr. Lal Patla, we have automated system for harvesting, automated system for slide preparation. So it was really making our life very easy. Because manually, if we continue and making the slides or do the harvesting, we may make mistakes because we get disturbed, or if we are not in a right state of mind, we may make mistake, we may add some wrong chemical. But these machines are really good. You just load the sample and walk away, and it will help you to finish the case. And then you take those slides which is prepared by the machine and put in the robotic microscope. It will scan the slides completely within few seconds, where manually we take 15 to 20 minutes, it scans in 5 minutes and keep it ready for analysis. That is where I would say the cytogenetics advancement has gone very good, and it is helping especially to the labs or the companies who are dealing with lots of sample. Of course, in Dubai we do not have that much sample which I was dealing in India. In India, I would say I was dealing every day almost 100 samples in cytogenetics, which is quite large. And doing that manually is very big challenge for us, even if you have lots of staff, 15, 20 staff with you, but still it is very big challenge. But implementing this automation, as far as I remember, the Japanese company is only making Hanabi is the name of the company who is making this automation, and that is the best advancement I would say which has happened in the cytogenetics field, really.
Thank you. I mean, that's amazing to know like that AI has stepped into cytogenetics as well, I think. And I think with time, maybe, if the algorithm gets trained, and as you rightly said, that it's not about replacing any human being, that's not possible, at least not in any
Yeah,
field, and obviously not in cytogenetics. We really need the human eye and, brain to predict. But I think, as you rightly said, automation always makes our life easier in terms of productivity, or it makes the analysis faster. So
yeah,
that's very true, and I think it's promising times ahead with AI and integration and automation and all those stuff. So
right.
uh, but I'm sure, I mean, I have been like as a student or later on also, I have made my hands dirty in culturing cells and looking like karyotyping. So I know there are a lot of challenges in this field. So and particularly when you are working in terms of in the field of medical diagnostics using these high end technology. So would you like to share some of the biggest challenges that you have faced, or you think that these challenges could be, um, opportunities for the future, for the development of the field?
Yeah. So the biggest challenge what we face in cytogenetics is getting a trained manpower. Trained manpower is the biggest challenge because not everyone can handle cytogenetics samples, because you need lots of expertise. If I make slides, I make a different way, the other person will make in a different way, but the software or the person who is reading the slides later on, and they are getting a different types of preparation, and which makes their life also very difficult. So that's where I would say the training of cytogenetics is very important, and that's where the person should spend almost 3 to 4 months. If they are very basic, they don't have any knowledge about cytogenetics, 3 to 4 months training is must required to get hands-on training and proper expertise in making slides. And as we know that expertise will come definitely after years and years of practice and everything. But as a technologist who are doing the test, or who is preparing the slides and everything, culturing and all, they must be trained 3 to 4 months. That is the biggest challenge. And in our country, unlike in Western countries like America, or if you see the European countries, they have a particular curriculum for that cytogenetics. So everyone needs to go for that training and everything. And if they want to pursue their career, they go for further training or fellowship, and they get very good people there. So there is no problem with the manpower in these countries. But in spite of so much of big population in our country and so many graduates are passing out every year, I see that lots of people are still not aware of the cytogenetics. And I would urge youngsters, actually, if you really want to pursue your career in a very nice field, and if you are a science student and you want to do something in the lab side, cytogenetics is the test really will help you to grow in the career. And the training is important. This is the biggest challenge. Second challenge is getting a supplier in time, because the chemicals, or for example, I use a particular media for my culture, and I'm very comfortable using that media for kind of a long time, and then suddenly the supplier close that media or stop the media or something, then again I have to take another media and do the validation, do the quality check and everything. It's kind of a big challenge for us. This is really really big challenge. It sounds very normal thing, but really a big challenge for the cytogeneticists who are really doing the culture and everything, because every media has their own quality. You will not get a proper chromosomes, then you have to break your head, do the reculturing, finding out the problem. pH goes up, pH goes down, the nutrients are not okay, it is not stored in a proper temperature. All those challenges are really there. And especially in cytogenetics, the quality of chromosome is very important again. So if you do the culture and you don't get a good quality chromosomes, you will miss lots of abnormalities which is there. So our challenge is to get the longer chromosomes as much as we can, so that we do not miss at least gross abnormality less than 1 MB. More than 1 MB we should pick up in cytogenetics, but less than 1 MB, of course, it is very difficult for a human eye to pick up. So these are the things I would say are challenges: getting proper trained staff, and the suppliers, and then the process technology side, that we have to get a good quality chromosomes. So these are the challenges I feel are there. Yeah.
Yeah, I think you told the very practical problems, and I think with time, probably, these things would, we might see more brighter days in the time ahead. And you rightly said, like, because even I have seen that particularly people or students who are graduating or doing the post graduations, even they are confused from the even from the fields of biotechnology, genetics, cytogenetics, specialty. I also personally feel, I mean, from academician's mind, I say that we need to speak about it more. We need to make them aware, and definitely the training, it is extremely important. It is
Yes.
extremely important, like you rightly said. So just that sort of from your previous insightful sharing that you were talking about in terms of infertility and stuff. Is there a connection between cytogenetics field and genetic counseling? I mean, is there a bridge or is there a gap? That is my question. Yeah.
There is definitely a very strong bridge between these two. How is it important? I will explain. So, particularly in the field of cytogenetics, with respect to infertility or recurrent abortion. Okay, the first thing comes in mind, that if for the patient point of view, that whether we can have a normal baby in future or no, or what we should do, because that chromosome abnormality is there in our germline cells. We cannot take it out, we cannot repair it, we cannot do anything. So what we need to do, so here genetic counselor is very important. So genetic counselor is the person who has a very nice knowledge about the subject, about the genetics, and they know how to explain the problems to the patient in their layman language, right? Because they cannot talk that, okay, this gene is not there, that gene is not there, this is the translocation. They will not understand. The genetic counselor is very, very important here, making a very strong bridge between the laboratory or the doctor with the patient. So with the patient, they will explain that we have found this in your cells or in your body. What we need to do is, now you have to think about that there is a 50% chance or there is a 25% chance he will make a pedigree of the family. If there is a family history, if something is running in the family, they will explain accordingly that there are chances of getting abnormal baby again, or if you go like this, you may have a normal baby as well. Or you have to take, in case of cancers, it is very important for patient also to understand now that what type of abnormality we have, which is good prognosis, worse prognosis, or what is it. Of course, in cancer, most of the time the oncologist will always explain that what is it, but sometimes oncologists also need a help of genetic counselor to explain to the patient, especially in the case of sequencing, exome sequencing, whole exome sequencing, or NGS based tests, because there are multiple genes which are available in your plate and you don't know what to do, what to say. So genetic counselor will help a patient to understand that this is the thing we have found and what we need to do. So definitely genetic counselor is a very strong bridge between clinicians, a laboratory scientist or laboratory personnel with the patient. Yeah.
Thank you, Saurabh. It's very nice talking to you, and we are almost at the concluding part of our conversation. Though I am very excited, and I really not want to let you go, and I can actually talk hours together on genetics and cytogenetics. But keeping a line of the time, so. I think you already gave some aspects when you were talking about the challenges that you face, and I mean that we all face in this field. So I would like to ask you that what would be your message to aspiring scientists, or students from the field of biology, genetics, or like life sciences as per in a broader umbrella?
Yeah.
Um, how can they get into this field, and like you have an extensive and very rich career, and today you are sitting at the one of the topmost chairs of organization. So can you give some tips of if somebody wants to follow a career path like you, what would be that?
Yeah, this is the best part for me to explain, because I want lots and lots of people to come in this field, and how they choose the career, how they, not only cytogenetics, I would say, if they really want to pursue the career in molecular field or cytogenetics or biochemical genetics, because biochemical genetics is also very important now in the lab diagnostics, right? Newborn screening, and then thalassemia or different types of genetics are really, really important to detect through biochemical genetic tests, through LCMS, MS, and GCMS, and all those things. Same molecular biology. If they want to pursue the career in molecular biology, there are lots of laboratory importance is there in molecular genetics, because they can do the sequencing, they can do the PCR, real-time PCRs, qPCR. So many molecular-based tests are there, which we are doing here also. So my message to all the aspirants and all the youngsters is, follow the passion. Whatever you want to do it from your heart and continue doing it. Do not stop, because God will help you. And if you really want to do that, nobody can stop you. Even the financial constraints are there in life, the family pressures are there. Sometimes parents are also thinking that why my kid is not earning now, why he is always wanted to do. I would say uh, don't stop anywhere. Continue with this. One day you will see that you are reaching in the position which you really dream of, if you really follow your passion. In field of cytogenetics, there are very few institutions in India who are giving a proper degree, and especially in South India, we will find out in Bangalore, in of course Christian Medical College, Vellore Institute of Technology, and Andhra University. Andhra University is one of the pioneer institutions in India for cytogenetics, because I know lots of geneticists who have passed out and they are very, very well placed. And lots of my teachers are all from South India, Andhra University, and Tamil Nadu, MGR University, and so on. So, if you want to pursue your genetics career in the best way, I would suggest to go to the South India and get your degree, and then decide definitely that you want to follow cytogenetics or molecular genetics or biochemical genetics. Get the industrial training, go to the companies, try to convince them. It is very hard to get into the companies and industries, but try to convince them. Even if you have to pay from your pocket, get the training, because you are going to learn something you will learn, and then you will use in your career. And people are thinking that if you go abroad and get the training only, it will help. No. If you see outside India, I've been to Mayo Clinic, I have been to different universities like Boston and everywhere. All the people who are working in the higher position in genetics are Indians. If you see MD Anderson, if you see MD Anderson, which is the father of cancer hospital, I would say the best
Yeah,
cancer hospital in the world,
right.
all all Indians is sitting in the biggest position, and they are doing it, cytogenetics or molecular or everything. Lots of my friends are still there and working. So follow the passion, do best, and think about if you really want to do, nobody can stop you. That's what my message to all the youngsters and aspirants. That's it. And any help from my side, I'm always open to help everyone. No problem.
Yeah, I
Yeah.
think once I mean everybody who will be who's watching this at this point, or who will be watching it later, I think they can they connect, can you tell where they can connect you,
Yeah,
if
my
it's
best way
possible?
to connect is the LinkedIn page. So my LinkedIn is there in the name of Saurabh Bhattacharya. They can find me there, and they can connect to me and ask me questions. Of course, I cannot offer jobs to everyone, but I can actually help them to understand what they want to do in career, or if they really want my reference or recommendations. I'm always open to help people. So they can connect me through LinkedIn.
Amazing. Thank you so much, Shora, for your time. And I think we had a very fantastic, at least for me, it was very exciting. And I'm burning with, excitement and the kind of insights we all got from you. Thank you so much. And thank you so much for giving so much time on
Thank
this
you. Thank you.
podcast.
My pleasure. Thank you. Nice talking here.
Yeah. Thank you, everybody, for watching this. And we will have more exciting episodes. Keep watching Eclectica and subscribe or follow me on LinkedIn and subscribe my YouTube channel for the same. Thank you. Bye bye.